Hello students, today we are going to start our second botany class. As in previous class we studied about reproduction in organisms introduction and also studied about reproduction, and the definition of reproduction also says that it is a biological process in which living organisms produces a copy of its own kind. So we also studied that it is a continuous process that a species transfer from a generation to generation also, and further it is divided into two types. One is your asexual reproduction, another one is your sexual reproduction. So in this features of asexual reproduction we also studied about the types such as your binary fission and multiple fission, budding, gamule, fragmentation, vegetative propagation. So today we are going to elaborate all the parts serially, like first binary fission, next one is your multiple fission, then budding, then gamule. These are quite important in your examination point of view, as some of the short notes of this topic. So we start our study firstly in the binary fission, as it's denoted that the term binary means two. So it is a common and rapid method of reproduction in lower organisms which occur in bacteria and your amoeba, and somehow in other protozoans also. So these types of conditions are takes place during favorable conditions. The parent divided into two equal and identical halves. As we saw that it is a sequential process. This is the first organism, and when it started dividing, its nucleus is started splitting, and as the cell materials and cell membrane and cell materials also started dividing, in some stages it is divided, its nucleus also divided into two types, and finally it is gives rise to two fission process. It is the splitting, the elaborate and the splitting process is otherwise known as your binary fission. So it is a mitotic division which you studied in your previous cell division chapters also. So in this mitotic division, according to the theory of mitotic division, it is a division process which is very simple, and it is divided into three types of binary fissions in binary fissions, which are you can study it as a simple binary fission, transverse binary fission, and longitudinal binary fission. It is divided into three types. One is your simple, second one is your transverse, and third one is your longitudinal. That means it is your simple binary fission which we studied about in this diagram. This is the nucleus. Here the nucleus started dividing, and then the nucleus fully splitted. This is the mitotic division in which the elaboration takes place of the particle, and then the nucleus is divided into two types. Here the nucleus 1, here the nucleus 2. It is the fully structure of the binary fission. Here two particles are separated. So this is the features of your binary fissions. So the simple binary fission, transverse binary fission, and longitudinal binary fission also occur three types and three types of stages also. So in simple binary fission we see this in regular amoeba. This type of binary fission takes place in amoeba, so it is an example of amoeba. Then transverse binary fission, it is transversely, or it is divided in a long axis. This is the simple process, but the process is same, but the division takes place in a single long axis. In the other hand, the longitudinal binary fission means it takes place in protozoans, which takes in amoeba and protozoans, because the binary fission also takes place in protozoans, bacteria, amoeba. So it is the longitudinal transverse process. Longitudinal binary fission also takes place in flagellated protozoans, such like euglena. Her body also divided into longitudinal main axis. So it is all about your binary fission. We again repeat that this type of process takes place in bacteria, amoeba and other protozoans. It is a single sequential process in which nucleus is fully divided into two types. So in this process, nucleus is started to divide in mitotic process. It is a mitotic division. It takes place. Simple binary fission takes place in amoeba. It is orderly divided into three types. One is your simple binary fission, another one is your transverse binary fission, next one is your longitudinal binary fission. So simple binary fission means your simple division process in which nucleus is divided into two types. So in this process, mitotic division takes place. In transverse division also mitotic division takes place, and longitudinal division also mitotic division takes place. It is same in all asexual reproduction and its types. So it is all about your binary fission. Nextly, we are going to study about your multiple fission. Okay, nextly we are going to study about your multiple fission. As we previously studied about our binary fission, and this term says that bi means two, and here we wrote about multiple fission. Here multi means a number of. In previously we studied that the organ is the amoeba is divided into splitting into two, but here we studied about multiple fission, and here it's divided into a number of parts. It's going to divide into number of daughter cells which are released from this host cell. So in this part we going to know that in which organisms mostly the multiple fissions occur. Mostly it's occur in your algae, fungi and protozoans. So it is also a most common process of your asexual reproduction which takes place in these organisms. So in this method we going to know that a single parent body is produce many daughter organisms. That means a single parent body, here is the single parent body, it is going to reduces a number of daughter cells in the last stage. So it is, at first we know that the outer cell of this organ is covered with three layers, which is otherwise known as your cyst, and it is on this phenomenon is otherwise called as your encystment. And after encystment, the cyst is consist of a host cell and some of your cytoplasm and nucleus also. Further going to the second step, the cell is going to divide it into, the nucleus is going to divide it into number of parts. A number of nucleus are produced from a single nucleus. That means a nucleus is divided rapidly, and the rapid division of the nucleus is otherwise known as your karyokinesis, which is takes place in your second step of this multiple fission. In this karyokinesis process, a large number of daughter nuclear process, here is the daughter nucleus. The nucleus started, the cyst wall formed in the first stage, and the cyst wall going to divide the nucleus in your second stage. The division process of this nucleus is otherwise known as your karyokinesis, and from the karyokinesis your production of daughter nucleus takes place. I hope it is clear to all of you. Then we are going to the last step. On favorable condition, it is going to rupture, but from previously each daughter nuclei is surrounded by some number of cytoplasmic cytoplasms also. As the nucleus going to divide in this stage, it also the nucleus also consist of a number of cytoplasmic liquids also in it, because it is quite gelatinous in nature. So it is going to surrounded, the nucleus is going to surrounded by some types of cytoplasm with it. Then it going to rupture as a number of particles are gathering in it in favorable conditions. It going to rupture as the cyst wall ruptures and the spores released to give a number of new individuals, which is otherwisely known as your daughter cells. Again I repeat, it is your first cell in which the formation of 3-layered cyst formation takes place in your first stage. Then the division of the nucleus takes place, and first nucleus takes place, which is otherwisely known as your karyokinesis process. In the karyokinesis process, nucleus is divided and forms a number of daughter nucleus. As in the previous stage, nucleus is consist of a number of cytoplasm content in it. So here also the nucleus is going to contain a number of cytoplasmic liquids in it. So it's going to attach with a number of gelatinous cytoplasmic nutrient mediums, and after some time, when the favorable conditions appear, it's going to rupture and the daughter cells are going to released outside. It is all about your multiple fission. Nextly we are going to study about budding. Nextly we are going to start our third short note, which is budding. Budding means a bud, which this is a common type of reproduction which takes place mostly in yeast and hydra. Actually this is the diagram of your yeast, budding of yeast, but hydra is the diagram of hydra is also given in your book of NCERT. Somehow like this structure is going to some flagellated structure in your outgrowth, which is takes place in a surface, and laterally it gives rise to a daughter hydra. So later we will discuss about your hydra. So firstly we focus that it is these types of reproduction, just like budding takes place in favorable condition, in which body wall bulks out. Bulks out means it is oozes out. It's a bud like structure takes place in its outside, and firstly here the nucleus division takes place. In the previous means in your multiple fission we studied a point that in the case of your nucleus division, which takes place which repeatedly divided to form your karyokinesis process, that the formation of nuclei takes place. Such like that here also the mitotic division takes place, but here the nucleus is going to divided into two types. Nucleus is going to divided into two, number of two. Here the nucleus, here the bud, actually here the bud is here the outgrowth is takes place, here the bud is takes place, but in initial stage the nucleus is splitting out, and the nucleus is going to divided and some nutrient medium also with it. In repeatedly it is going to divide and redivide to form a number of buds. Actually it is not a single type, it's also going to divide a number of buds in it, just like your pseudomycelia stages. It is a chain buds is called your pseudomycelia. So it is a mitotic process which forms two nuclei, moves into a bud along with your cytoplasm. Now the bud is enlarges, somehow it is enlarges, its size is going to larger and larger, and somehow it is detached from its parent body. But in this case of your hydra is going to release its outgrowth in its parent body. After detached of its parent body, the daughter nuclei is going to detach from the parent body. It is quite different from the budding stage, but both are the examples of your budding. Nextly we are going to study about our we are going to study about our next short note, which is your gamule. I want to clear about gamule, that in the stages of gamule, uh, it is a interesting term in it, that the formation of sponges, it is nothing but in autumn, and in autumn freshwater sponges suffering cold and food scarcity actually, and it die and disintegrate, and leaving behind a large number of gamules. Uh, these are this is the formation of gamule, and it is a structure of your spongy, freshwater spongy, which is going to reproduce a number of gamules in it. Here we saw that the gamule is, here is the diagram of your gamule. So as you saw the diagram, today I am recording a single hand, so it is quite problem to recording the video, please adjust in it. So the structure of your gamule is consist of a number of parts. First we know the parts, how they are formed. Actually this gamule is consist of a number of parts, and its outer parts is known as, it is its mouth parts, and the opening is otherwisely known as your micropyle. There is some types of tentacle like structures which is otherwisely known as your spicules, and the inner mass, the rounded structures, other mass of the, it is the main constituent of this spongy or this gamule structure. So this mass, this all mass is overally known as your archaeocytes, and the outer surface, the green color circle, is otherwisely known as your, this blue to this green, this all circle is otherwisely known as your gelatinous material. Gelatinous material, it is all about your leveling of this diagram. So now we know about the characteristics feature of this gamule. Actually during unfavorable conditions, these types of structures are actually formed in freshwater sponges, and some marine sponges are also developed in this same process. It's going to form the internal boards. So the internal boards actually the gamules, and it forms to furtherly, it forms to the spongy. So gamules being when small group of cells called archaeocytes. Archaeocytes means this is a small group of gamules. These are the gamules. Actually they are oozes out in the favorable conditions. It is otherwise known as your archaeocytes, which are laden. The reserve food granules become isolated from internal surface of spongy. It is going to these archaeocytes are in the unfavorable conditions inside the inside the gemule, and it's covered a mass, it is a mass of cells, a mass of other cells are also dead cells are also, because there is a scarcity of foods takes place in it, so some types of dead cells are also produced in it, and number of lively cells are also covered in it, covered in the gemule. Its mass is covered by a double layer and refined spicules, and it is covered by a gelatinous material from the extraction and oozes out of the archaeocytes actually. So it is covered by a gelatinous surface medium for the safety of these archaeocytes, and for further safety also, it is some types of tentacles are also present in the outer surface, which is otherwise known as your spicules, and it is takes place in, it is the these types of structure are placed in your autumn season, as in starting in the starting phase we are going to I told you about that. That means the gamules are not formed in the winter conditions. When the springs comes, the gamules become hatches out, that means from the microplasmids, that means it is the opening. Here it is the opening, and from this side, the micropellar end side, the gamules are going to produce it, the micropellars, the archaeocytes are going to out from this stage and escape through the micropellar, develop a number of new sponges. Actually it is going to have a number of gamules are produced in a single surface, so they are going to oozes out and forms the number of sponges in their outer cells. When favorable conditions arise, the gamules germinate and going to their other growing phases also. You just know about the some types of features and some types of labelings of these diagrams, then we follow the notes. You can know all about this gamule.